All right, welcome to my first ed puzzle in biology, chapter 1, the science of biology, specifically looking at section 1, which is titled, What is Science? So we do have a couple learning objectives for today, and that is describe the goals of science, explain the procedures that are at the core of scientific methodology, basically the scientific method, and then define what a scientific theory is. So science is basically the use of evidence to construct testable explanations and predictions of natural phenomena and to gain knowledge. So it's basically studying the world around you. You're using evidence to explain the natural phenomena that you're seeing. Facts and ideas can change due to new information that can arise, and this is true. I'll give you some examples when we get to like the cell theory. Science is a very fluid process. I know maybe in elementary school you were taught the scientific method and how it was very rigid. Like first you do this, you ask a question, then you test it, and then you collect data. Well, that's not always the case. It's actually more like a flow chart, and depending on the situation, you can jump from one concept to another. But it does deal with only the natural world. A lot of times we organize our information to look for patterns or trends, and then the explanations that we come up with are based on the evidence that we've collected in those experiments. Now, the goals of science is to obviously try to explain the natural phenomena that we see. Okay, so to provide natural and testable explanations. So we can test these explanations to verify, yeah, that's what's going on here. And then we try to use these explanations that's supported by the data that we're collecting to hopefully make predictions about the future or to see patterns. Now, I know this last statement here is kind of trippy, but it says every time we make a scientific discovery, more questions are raised than answered, because now we go in deeper, and sometimes we tend to go into other fields when we discover new discoveries. But the thing is, I really want you to question everything, even in this class. Science never proves anything, it can only really disprove. That's the trippy statement. It never proves anything, it can only disprove a statement. So we can accept the hypothesis, but know that it can be rejected later on if new evidence should arise. So really the only thing we can ever do in science is to disprove something. And if you hear my son in the background, he's chatting it up. All right, so scientific methodology involves, first off, observing. Okay, you make an observation of something and then you ask questions. It's the act of noticing and describing events. A lot of times you tend to use the five senses, and you may organize these observations in an orderly way. And then you're going to form a hypothesis, which is an explanation that can be tested by an experiment. Oftentimes, hypothesis and inferences kind of go hand in hand, but an inference is a logical interpretation based on what you already know. So we're going to go through examples of observation versus inference here. So observations like what you can see, like you notice with your five senses, and an inference is, if I go back, a logical interpretation. So it says the dinosaurs were fighting because all the fossilized footprints were jumbled up together. Well, the observation is, what did you notice? So you can say, okay, I noticed that the fossilized footprints were all jumbled up together. Okay, so that would be your observation. And your inference would be like, well, maybe they were fighting. And that's why they're all jumbled up together. The sky was changing colors from dark to light because the sun is rising. So, what's the observation here? Hopefully, you're saying that the sky is changing colors, and then the inference, the sun is rising. Jenny's mom burned dinner downstairs, and Jenny could hear the smoke detector upstairs. Her observation is the smoke detector, and then her inference, burnt dinner. The kitchen is warmer inside than inside the freezer. So Emmanuel Lewis's ice cubes were melting on the counter. So he noticed that his ice cubes were melting. That is your observation, and his inference is that it must be warmer inside than inside the freezer. All right, conducting controlled experiments. So when we test our hypotheses, okay, statements that can be tested, there's a couple things you need to be aware of. First off, variables. So variables are conditions that can change in an experiment, and there's two types of variables that are extremely important. The first one is independent variable, what you or I change, what you manipulate in the experiment. Okay, so what are you changing in this experiment? If you're looking at plant height and the effects of light, so what you're changing is light, the amount of light a plant might receive. Okay, so that's your independent variable. That's what you change, what you manipulate. Your dependent variable is how you're going to measure it to see if what you change actually has an effect. So going back to how does light affect plant growth? Well, you're changing the light conditions. Maybe you're going to put a plant in a dark cabinet and a plant out on the windowsill. So that's your independent variable. Your dependent variable is what you measure. So maybe you're going to look at germination. Does the seed produce a plant? Or maybe you're going to measure it by plant height. But either way, make sure you get independent variables and dependent, like you understand the difference between those two. And then you're going to need a control group, a group that does not receive treatment. It's used as a benchmark that you can compare back to. So in my case of how does light affect plant growth, I'm going to have a plant in the cabinet. I'm going to have a plant on the windowsill, and then maybe I'm going to expose a plant to 24 hours of light. So my control group in this case would be the plant that's on the windowsill, because that's kind of like normal conditions. It's getting the amount of sunlight that the sun is up, during the day. So that would be my control group, and I would compare my 24 hours of light and my 24 hours of dark to it to see the results and to compare them and make inferences from and collect my data. So independent variables, what you manipulate, change the dependent variable. Okay, so this is just a summary of independent variable, the one thing you can change. And it's very important that in an experiment, you only change one thing. So in this experiment, just based on it, you can see that we have the same pot, the same type of plant. Hopefully you're using the same amount of soil, the same plant species, you're measuring it the same way. But the only thing that you're changing looks to me the liquid that you're using to water your plant. So in this case, soda, juice, and water. Okay, so that would be your independent variable. That's what you changed. Your dependent variable is what you measure, the change that happens because of the independent variable. So in this case, the height or the health of the plant. And then finally, the control is everything that pretty much remains constant. So the type of plant you used, the pot size itself, the amount of liquid. So I'm putting 20 mils of soda. That means 20 mils of juice and 20 mils of water, and so on and so forth. So everything else has to stay the same, and the only thing you're changing is the liquid. So here's another example, the effect of biofertilizer X on plant growth. So independent variable, in this case, we're going to apply fertilizer X in this experiment. That would be your independent variable. That's what you change. That's what you manipulate. Your dependent variable is plant growth, or maybe you're going to count the number of leaves, or maybe you're going to count the number of fruits. But whatever you determine to see if fertilizer X has an effect on the plant. Now my question to you is, what would be the control? So remember, the control is what's going to stay the same between these two. So hopefully you say like the same plant species, the same amount of water, the same soil, the same amount of light. Okay. All right, let's try to identify the independent and dependent variables in these examples. How do different amounts of sunlight affect plant growth? So what is the independent variable, the thing that you change? I hope you say different amounts of sunlight. And how you're going to measure it is plant growth. Moving on to another example. How do different amounts of water affect plant growth? So what's the thing that you change in this experiment? Different amounts of water. What's the thing that you're going to measure to see if the independent variable has an effect on the plant? In this case, plant growth. How does height affect crater impacts? So what's the thing you're going to change? Height. So maybe you're going to drop a ball from 20 centimeters, 30 centimeters, 40 centimeters, so on and so forth. And then how are you going to measure the impact of that? That would be your dependent variable. You'd measure the diameter of crater impacts. Okay. So a couple years ago, the Herman Fair, okay, I shouldn't say the Herman Fair, it's the Grant County Fair, um, put on a display called the Whitey Tidy Experiment, basically looking at bacteria microbes in the soil of tilled farm fields versus no-till. So what this farmer did was that they took cotton underwear and they buried it in the soil, leaving the waistband exposed. And they came back 3 weeks later to see what happened to the underwear and how much underwear remains after 3 weeks reflects the level of biological activity. In this case, if you hardly get any underwear left, that means you have really good biological activity. There's lots of microorganisms working on it and eating that cotton. So microorganisms need carbon, and when you till, you tend to disrupt the carbon in a very tornado-like fashion. No-till obviously has a little bit more microorganism activity. So you could say, based on this picture, that this pair of underwear has little biological activity in the soil, while this pair has several, and I would say this is a pretty good amount too. Okay. All right, now moving on to collecting and analyzing data. There's two types of data that you can collect in a scientific experiment, and that is quantitative, which means numbers. Okay, maybe you're going to count something, maybe you're going to measure something, but basically it has to revolve around numbers. And then you have qualitative, which is descriptive, and you tend to use the five senses and hopefully not the taste. But quantitative data, you're going to use rulers, balances, graduated cylinders, thermometers, anything that is measurable with numbers. And that qualitative data, you're going to use your five senses to describe the observation that you're seeing. We want to lean towards quantitative data. Okay, so we can do statistical analysis. This is just extra stuff, and I mean, yes, it's good, but you don't want to base your experiment solely on qualitative data. So here I have a can of Coke. What are some examples of quantitative data? So hopefully you can say the amount of fluid that's in it. 12 fluid ounces or 355 milliliters or 140 calories in a can. What are some examples of qualitative data? You can say that the can is made of a shiny metal. Maybe you know it's made of aluminum, that it's red and white, that it has letters on it. Okay. So the reason why we want to lean towards more quantitative data is so we can do statistical analysis. And statistical analysis can tell us if our data is significant or not significant. It means it tells us if it happened by chance or if you're really onto something here, that there's a pattern or a trend that needs to be examined. We also need to try to avoid sources of error by having a good sample size. So you just can't do an experiment once with just like one plant or well, if we use plants, one plant, you'd want to have multiple plants and you want to do multiple trials. Um, not every hypothesis that you come up with can be tested by experiments. Anytime you do an experiment with animal behaviors, there tends to be too many variables involved. And so with ecology, they might do an experimental field investigation, but maybe one also in a lab to try to control some of those variables. So here's a graph showing the number of slugs per container and the food consumption rate per centimeter squared. And you might see that these little bars right here, these are called error bars. And if you see an asterisk above an error bar, that means that this data is significant compared to the other data. A lot of times, if these error bars do not overlap, then it is significant. So you can see 1 and 3 are significant, but I think 1 and 5, they overlap, and so it's not significant. Some more examples. Honestly, I don't remember what SBR grade is, but you can see that we have little asterisks here to show that the data is significant. Okay. So finally, the scientific method in science is not step by step. It is a flow chart where you make observations, you might think of interesting questions, form a hypothesis that you can test. You'll develop and test those predictions. You'll gather data. Maybe you'll have to go back and reject your hypothesis and refine your experiment to make it a little bit better. And then eventually, those ideas might develop general theories of the phenomena that we see. Okay, so one last, you know what, I'm not going to have enough time here. So I'm going to stop right here, actually, and I'll probably just recap this. So we'll have to break section 1 into 2 parts. But hopefully you know what qualitative versus quantitative data is, independent variable versus dependent variable, and observation versus inference.